Great. Welcome, everyone. My name is Ida Fernandez, and I'm the Executive Director of the Imagine Research Network. Joining me today is Dr. Mike Surette. He is the lead investigator for the microbiome for the Imagine Network's main medical research study, MAGIC, which stands for Mind and Gut Interactions Cohort. Dr. Surette is a microbiologist from McMaster University. We're talking to Dr. Surette to get some information about the design of the study. Can you explain a little bit, how does the study work? And what would you describe as the purpose of the MAGIC study?
So the MAGIC study is a Canada-wide investigation looking at healthy individuals with inflammatory bowel disease and irritable bowel syndrome. And the idea is to collect lots of information in clinical samples to really characterize these individuals and understand as much as we can about disease phenotypes and diet and other measures of health, as well as collecting stool samples to study the microbiome. The study aims to collect up to samples from up to 8,000 people over multiple years to look both at the improvements in health or decreases in health and how we might be able to use that to correlate to changes in disease. And again, for us, how that interacts with the microbiome.
Great. So what is your role specifically in the MAGIC study?
So as coordinator of the microbiome component of this, my lab is fortunate enough to get all the stool samples sent to them. And so the first thing that we do is we actually collect the stool samples, process it, split it up into multiple samples that are stored long-term for studies later on as new questions arise or new methods are developed to analyze the stool. But our main role is actually to use the stool samples to understand the microbiome. And so we extract DNA and use that DNA to characterize the microbiome.
Can you describe what is the microbiome?
So the microbiome is the collection of microbes that live on or in us. And although most people think of microbes as infectious agents, we are actually a walking ecosystem of bacteria and other microbes. And in fact, they are essential and critical for human health, as well as playing roles in disease. And so there's a couple of important things about the microbiome. Most of it is, in terms of sheer number, is in the human gut. And within our gut, we have on the order of 1,000 different species of bacteria, and probably 100 to 1,000 more bacterial genes than human genes. So if you think of the 25,000 genes that it requires to make us, there are millions of bacterial genes that make up that microbial community. So it's very complex. The other issue is that we all have our own unique microbiome. And although they all perform similar functions, the microbiome that I have and the microbiome that you have are distinct. And this individual variability and difference from individual to individual is a good thing in many respects, but it also means that we need to study large numbers of individuals to be able to tell the microbiome from a healthy person from a diseased person. And that's one of the reasons why the MAGIC cohort is so big.
So what information can we learn from the stool samples?
So, the stool is really quite a wealth of information. And from the microbiology perspective, the microbiome sits at a really important interface of us and the outside world. And first off, anything that we eat, our diet, the first thing that they encounter is microbes. And the microbes play a really critical role in diet. And so many foods that we can process and digest are processed and digested by the microbes, and they produce products that we can use. And one of these that you may hear about is the short chain fatty acids, which are really important. And this is why we're supposed to eat fiber. And we can't digest fiber, but the microbes digest fiber and convert it to short chain fatty acids. They also make essential vitamins that we need. And the other critical role that they play is with the immune system. So over half of our immune system cells are in the gut. And that interface between the microbiome and the immune system really both trains and maintains the immune system throughout life. And where this balance between the microbiome and the immune system is perturbed, that can lead to chronic disorders and inflammatory diseases such as inflammatory bowel disease and to some extent irritable bowel syndrome. So the microbiome really sits at this critical interface between us and the rest of the world. And the microbiome, as we learn more about it, will be able to tell us what critical diet, what are the right diet components for me? What are the right diet components for you? Is my microbiome not interacting well with my immune system, and how can I correct that? So it's really at the interface of health and disease very broadly, and understanding this interaction, which again is very complex, is going to really open the doors to new avenues for managing disease.
So can you explain how the microbiome data help us better treat specifically IBS and IBD? I mean, what's your vision for treatment options?
So one of the things we do now, particularly with IBD, is we treat the symptoms. IBD is an inflammatory disease. We have an overt inflammatory reaction in the gut that causes lots of tissue damage. That's ultimately what causes disease. That's a reaction of the immune system to its environment, which is partly microbes. If you, instead of using anti-inflammatories to suppress disease, remove those triggers or alter the triggers, alter that balance between the immune system and the environment, which is mostly microbes, you can get at the essence of the disease and treat the cause of the disease rather than the symptoms. The challenge, of course, is that, again, because the microbiome is so variable, it's hard to predict those from studies with small numbers of individuals. But as we gather more and more data in these large studies like MAGIC, we'll start to be able to say, these are the pathways, these are the microbes that we can target and direct. And it may be some really relatively straightforward things. What is the best diet for my microbe in me, as opposed to what's the best diet for people with IBS, or what's the best diet with people with ulcerative colitis. So the microbiome is a way that we can really personalize medicine. And so you hear a lot about personalized medicine, but the microbiome is one of the most personalized aspects of our body. And again, that's one of the areas that a study like MAGIC will help us really fine tune our treatments to make them more specific towards individuals or groups of individuals that present the same disease in slightly different ways.
Why is it important to collect stool samples from participants for so many years?
Well, the value is that a single snapshot in time only tells you so much. And that's true of any kind of data that you ask of an individual. You can take someone's blood pressure on one day, happens to be a day that their blood pressure is a little elevated. It's really not the picture. So the microbiome does change over time. It is relatively stable, which is a good thing. But of course, disease changes over time. And so if an individual with IBS develops worse disease or shows improvements, we can start to correlate that changes in disease or changes in health with changes in the microbiome. And that gives us much more power to be able to start to decipher this interaction between the microbiome and us.